Welcome to Computational Genome Biology YouTube channel. Large language models like GPT function effectively because human text is naturally tokenized. In English, sentences separate into discrete blocks, words, using spaces and punctuation. DNA, however, is a continuous, unbroken string of billions of letters with no such delimiters. To force this code into a model, previous researchers relied on rigid structures. They either processed DNA one single letter at a time or mechanically chopped the sequence into rigid, perfectly equal blocks, a method known as k-mers. Biological instructions ignore fixed grids. When a specific sequence, like a protein binding site, straddles the edge of a chunk, the fixed cutting mechanism slices that functional biological word right in half, destroying its meaning. These fractures compromise the model's ability to generalize across different tasks. It also makes findings nearly impossible to interpret, as the resulting tokens rarely correspond to actual biological units. Without the ability to recognize where a biological instruction begins or ends, a model operates on blind statistical guesses. Precise decoding requires an algorithm that adapts to the native structure of the genome. GenART solves this structural bottleneck by dynamically segmenting raw DNA strands into variable-length, biologically meaningful words, rather than using rigid cut points. The adaptive tokenizer uses parallel 1D convolutional neural networks to extract local motifs. A multi-layer perceptron then scores every interbase position to calculate boundary likelihoods. Finally, a Gumbel softmax layer makes a binary decision: merge the bases or split into a new token. This mechanism completely removes the need for human annotation. The neural network organically deduces the native syntax of life directly from the raw sequence. This structural precision leads to an efficiency paradox when GenART is tested against the industry titans. This chart compares average performance across 33 genomic tasks. Despite operating with only 350 million to 1 billion parameters, GenART models match or defeat 7 billion parameter models like EVO2 across almost every category. This dominance is particularly clear in long context challenges. In the 10,000 nucleotide splice AI benchmark, GenART maintains high accuracy where larger models lose the signal. These benchmark victories strongly suggest that intelligent architectural precision can effectively overcome the sheer need for brute force parameter scaling. Beyond raw scores, the most significant finding is what GenART's internal representations actually learned about biological reality. This mapping reveals a common failure. In standard models, data points for enhancers and splice sites are hopelessly tangled together. But GenART's adaptive approach relies on contextual signals, naturally separating these functions into clear, discrete, color-coded clusters. These results indicate that GenART effectively reverse engineers the functional grammar of the genome, identifying specific biological roles solely from sequence context. The researchers also found that phylogenetic diversity acts as a powerful regularizer during pre-training. Training on multiple species prevents the model from overfitting to human-specific biases, fostering feature representations that apply across evolutionarily distant taxa. Paradoxically, as the architecture scaled to 1 billion parameters, the model chose to utilize much finer sequence tokens. A moment of wonder. This shift occurs because the 1 billion parameter model possesses a globally smoothed attention mechanism. Instead of relying on rigid, pre-assembled chunks, the larger architecture is capable of viewing the entire sequence holistically to integrate high-resolution details. GenART demonstrates that to decode the instruction manual of life, the shape of the algorithm must adapt to the function of biology. Thanks for watching. Please like and subscribe Computational Genome Biology YouTube channel.